Master in Anatomy and Physiology with Contd.
Welcome back, guys. In today's lecture, I'm going to talk about Endoplasmic Reticulum and Golgi Complex, which are the other two types of cellular organelles which are present in eukaryotic cells. And as you know that in last lecture we talked about endosymbiotics theory, in which we discussed about how mitochondria and chloroplast end up having in eukaryotic cells. In today's lecture, we're going to talk about two very important cellular organelles, endoplasmic reticulum and the Golgi complex, which are the part of endomembrane system, and we'll focus on the structure and the function of each. And we combined these two cellular organelles together because they actually work together, and it will be easier for us to understand their structural and the functional difference, and how actually they help each other during the cellular activities. So that's our target. So let's start by looking the structure of the cell. As you can see here in the picture, this is the nucleus, and with the nucleus there is a nuclear envelope through which another structure is attached. This is called the endoplasmic reticulum, and there are you can see there are rough endoplasmic reticulum and smooth endoplasmic reticulum. These are the types of endoplasmic reticulum, and these are usually two types of endoplasmic reticulum. One is rough and another is smooth. And as you can see in the picture, it looks rough and it looks smooth. And why it is that? So we're gonna focus that in detail in a few moments. And after that, you can see this another structure close to the endoplasmic reticulum. This is called a Golgi complex, which has different sides on it. As you can see, the name is given here, and I'm going to focus this one in detail in short moment also. And you can see something is moving from endoplasmic reticulum, like kind of like named as a vesicles, to the Golgi complex. Okay, so I think there should be some kind of relation. You might think of that from this picture, right? So that will also I'll explain in short moment what is going on here. Okay, so from this picture what we have to know is the nucleus, endoplasmic reticulum, and the Golgi complex all are working together for something. Okay, that something we'll focus now. Okay, so let me move on to the next slide. Maybe you are like so excited to know about the process, what's going on here. Okay, so let's move on. So in our next slide, what you're gonna see is the endoplasmic reticulum. As I mentioned you in previous structure, there are two types of endoplasmic reticulum. One is called soft endoplasmic reticulum, which is here, and another is rough endoplasmic reticulum, which is given here. Why smooth endoplasmic reticulum is named as smooth? Because it does not have ribosome on its surface. It does not. Okay, but rough endoplasmic reticulum has ribosomes on it. You can see this small white type of structure here. Those are called ribosomes. And let me tell you that these ribosomes are attached to this endoplasmic reticulum by glycoproteins such as riboforin first and riboforin second. Okay, so those proteins are called what? Riboforin. Okay, riboforin first and riboforin second. Okay, these are the two glycoproteins. Remember that these are the glycoproteins. Okay, these glycoproteins allow ribosomes to attach to the endoplasmic reticulum. And keep in mind that these ribosomes are not facing toward the lumen of the uh, endoplasmic reticulum, but they are facing toward the cytoplasmic side. Okay, basically This is the cytoplasm, and these ribosomes are facing outward. Okay, they are not facing inward. They are not. And as you can see, this is structure, like this kind of structure, like we call it a cisternae, and these cisternaes are usually unbranched. Okay, but they do have ribosomes, but they are unbranched. Okay, so that's the idea we need to think of. Now, when we're talking about endoplasmic reticulum, we must think of what are the main components of endoplasmic reticulum. So the main components of endoplasmic reticulum are, okay, so the first one we call that is a cisternae, as I mentioned you, right? Cisternae, right? Another is called tubules, right? And the third one we call that is vesicles. Okay, so cisternae, as I mentioned you, these are sac-like structure, and these are usually unbranched, and ribosome is present. Tubules, there is no ribosome on them. Okay, and it depends. Okay, usually tubules are present in smooth endoplasmic reticulum, like look tubular structure. They do not have ribosome, but sometimes, it may happen. Okay, you may have sometimes ribosomes on them too, but and usually absent. And the vesicles basically are usually are round in shape, and they have different proteins or different types of things inside them, and they do help in transportation. The main job is to help in transportation. They store something and they transport. Okay, they transport the contents from endoplasmic reticulum to the Golgi complex. Okay, or from Golgi complex to endoplasmic reticulum. So we call this anterograde and the retrograde type of transport mechanism. We're not gonna focus in detail, like process that will be in the cell bio lecture, but we're just gonna talk like a little bit, like general structure what's going on. But from this picture, what you need to know is that two types of endoplasmic reticulum, smooth and the rough. And the smooth do not have ribosome on them. Rough, they do have ribosome. Smooth endoplasmic reticulum help in detoxification in the liver, and in steroid biosynthesis and calcium storage. In the case of muscle, is a sarcoplasmic reticulum. So sarcoplasmic reticulum is one of the modification of the endoplasmic reticulum. Other modifications include T-tubule and myeloid bodies. Okay, the T-tubules are also present in the muscle, and they help in the transmission of the stimulus of the contraction. Okay, and the myeloid bodies are present in retina, in eyes, and they help in the pigment migration. So there are three modifications that you can see from the endoplasmic reticulum, and those were sarcoplasmic reticulum, SR, and another is called T-tubule, right? And the final one we call that is a myeloid, okay, myeloid body. Okay, these are the three modification. Okay, three modification. Okay, so that's the general idea about endoplasmic reticulum. Now, when you talk about rough endoplasmic reticulum, what is the main function of rough endoplasmic reticulum? The main function of rough endoplasmic reticulum is to synthesize proteins. Okay, those proteins may be soluble or integral membrane proteins, or sometime, endoplasmic reticulum, like kind of like resident ER proteins can be synthesized here. Moreover, the endoplasmic reticulum also play a role in folding, protein folding, and glycosylation, adding sugars in proteins kind of scenario. Okay, so those are the main function of rough endoplasmic reticulum. Now I'm going to talk about the structure and the function of Golgi complex now. Sorry, before I move on, this is the structure that we just discussed. The three component of the endoplasmic reticulum. You can see cisternae, tubules, and the vesicles. Okay, so you can see the structure, and I already mentioned about this. This is just for additional reference. Okay, so just see how they look like. Okay, now finally we're going to talk about the Golgi complex, and the Golgi complex looks like a very interesting organelle, right? And it is firstly identified or firstly discovered by Camillo Golgi in 1898. Okay, 1898, the biologist. Okay, so his name I cannot pronounce maybe, but it looks like Camillo Golgi, I think. He firstly discovered the Golgi complex, and the main function of Golgi complex is to process. Okay, is to package, package, and deliver. Okay, so deliver. It works is like a like a FedEx. This FedEx usually, everything whatever FedEx does, the job of the Golgi complex is kind of similar like FedEx company, right? They usually do process, package, and deliver items. So basically, if you look the structure, it has three face or three sides. One is called cisternae, and medial cisternae, and the transcisternae. And the cis-cisternae, often known as a cis-Golgi network or CGN, and trans-Golgi network TGN, which is a transcisternae. And as you can see, the trans-Golgi network is disappearing, right? And because like due to the budding, okay, budding of the vesicles, and just like it's losing its membrane capability, right? So that's the idea. So to describe the structure of the Golgi complex, there were two models were proposed, and those two models were very very appreciated in scientific field. Those models were vesicular, vesicular transport model, um, and another one is called cisternal maturation model. Okay, and these are the things you will focus on cell biology. Okay, but I'll just mention what actually they mean. Okay, these are the two models, okay, that actually explain the structure of the Golgi complex. Vesicular transmode model actually it means like this transcisternae or trans Golgi network and the cis Golgi network always, and the medial cisternae, they always stationary. Okay, they do not change. The only thing is, okay, there is a, let's say, endoplasmic reticulum here, and the cis face is facing toward endoplasmic reticulum. Whatever is budding from here is a vesicle gonna go, and it's gonna go, and the vesicle will pass. It will not diffuse or anything. Okay, this is just the way of this is just like a highway. Okay, it is just providing a tunnel to pass the vesicle. Okay, there is no change in the transcisternae, there is no change in the medial cisternae, and there is no change in the cis cisternae. That's the idea of vesicular transport model. And another model is cisternal maturation model, in which the cis Golgi network become like matured into a medial cisternae, and it eventually matured into a trans cisternae or trans Golgi network, and eventually it's gonna disappear. And something other vesicles gonna go from here, and they're gonna fuse to the cis side, and they're gonna form the cis cisternae. So whatever the components like lipid components or protein components in the endoplasmic reticulum will go and form the cis Golgi network, and that will go into and change or mature into a medial cisternae and go to the trans cisternae or trans Golgi network. So this is how it works in the case of cisternal maturation model. And cisternal maturation model uh, basically means that these cisternaes are transient. Okay, they are like short lifespan. Okay, And to actually prove it was truth or not, or it is like actually supported or not, they did a series of experiments. What they did is they stopped the budding process of the ER, and there were no vesicle formation. And what they see is there is the Golgi complex was disappeared after that. So that means what it tells us that, okay, when there is no budding from the ER, and if the Golgi complex disappear, that means the ER, whatever the vesicles formed from the ER are making the Golgi network or Golgi complex. So these are the two organelle of the same format. Okay, so what I want to mention is like this Golgi complex is part of endoplasmic reticulum. It just moved from the ER, and it just like form a kind of like uh, uh, kind of like very important organelle that actually process, package, and deliver proteins and different lipid components in the vesicles. Like vesicles gonna go diffuse, and they're gonna go after the here. This vesicle is gonna go or targeted into a different area. That's the idea of a Golgi complex. And Golgi complex is usually a stacked shape. You can see here a disc-shaped sacs. And if you try to look this Golgi complex, you need a heavy, metal stained. You have to stain with the heavy metal so you can see the Golgi complex. Okay, so that's about the endoplasmic reticulum and the Golgi complex. They are important. Okay, and they are very important in cellular activity, and they do perform an extraordinary job, okay, in endomembrane system. And if you want to just know more about this one, you can refer like cell bio lectures from other area, right? You can read from books that will explain in like detail about this endomembrane system. Thank you for watching.
Please subscribe to see more videos regarding anatomy and physiology.